Hello everyone, my name is Rudra Vengurlekar and today I am going to present my project of reproducing figures from this paper. This paper is from American Journal of Physiology, Heart and Circulatory Physiology. The paper titles A Model for Human Ventricular Tissue. This project is a part of Introduction to Biomembrane course. So let's first understand what is ventricular arrhythmias. Ventricles are the two bottommost chambers of heart. These and arrhythmias stand for irregular heartbeats. So the heart tissue comprises of three main layers. Inner one is endocardium, middle one is myocardium, and outer one is epicardium. And the nerve cells are in the heart are responsible for transferring signal across the heart so that heart could properly pump blood throughout the circulatory system. So, whenever there is irregular heartbeat, heart may stop or the blood flow can reduce, which may be fatal in some cases. So, this paper tries to propose a mathematical model of human heart tissues so that this arrhythmias can be studied properly. So what we want from the model? So the model should be uh, should be for human heart tissues. So at the time when this paper got published, the most of the models which were proposed were taken from the experiments on animal tissues, and animal heart tissues are very different from human heart tissues in terms of concentrations and time constants of various ions. So uh, the and also it was not very easy to perform experiments on human heart. So instead of performing many experiments, a mathematical model of human heart could be very beneficial in understanding such arrhythmias uh, properly. So here we want a mathematical model of human heart tissue which could generate action potential, which could simulate major ionic current and calcium transients. It should, from where we can get the action potential duration and the duration between two action potential. The model should be able uh, to properly capture the wave propagation, the conduction velocity restitution part. At the same time, as we are studying the propagation of signal in heart, um, we need we need a 2D or 3D solver to get how the waves are propagating inside the heart. So the model should be computationally simple enough for spatial temporal simulations. So that what is there in the proposed model in this paper. So how is this model proposed? So the model tries to capture every major and minor ionic current present in the heart tissue. It is sodium, potassium, and various other ionic currents. So these ionic currents are individually expressed as a Hoxie kind of model equations where there is activation, deactivation, and so on. So from these current, we can get the membrane potential by modeling it as a resistance capacitance model. So here, the second equation shows the change in membrane potential as a function of currents, the current stimulus, capacitance of the cell membrane, and resistivities in different directions. So this model proposes an S1 S2 protocol. So what is S1 S2 protocol? So in S1 S2 protocol, of S1 beat S1 signal is provided for a fixed amount of number at a fixed frequency, and there is one irregular beat after the last S1 which may have different magnitude or different frequency. So that's how we can study how these irregularities in the heartbeat are propagating inside the heart. Um, Also, this model assumes isotropic and continuum assumptions as in both the directions, in x and y direction, the properties are assumed to be same, and continuum because this model neglects the the ionic variations in the individual cells and instead models it as a continuous variation. To integrate this and many other ionic current equations forward Euler method is used along with Rush-Larson method. So I took the equations from the paper and I tried to simulate all the model parameters. So the few starting figures will be for different parameter values, how the parameter values are varying with potential. So this is the activation in with change in voltage. So this is figure 1a as shown here. So I simulated this figure. So it is activation as with increasing voltage it is increasing. This is the inactivation and it is also matching clearly. These are the time scales, the time constant for activation. It shows an increase and thereafter it decreases. This is the inactivation time scale. It also matches very clearly, and we can see there is a small change in slope here, which is also captured here properly. This is another time scale for parameter J. These are the normalized sodium currents, which are also matching properly at different membrane potential and impulse intervals. This is the normalized steady state sodium current which we saw in the previous slide as a function of frequency at two different temperatures at 37 and 21 degrees Celsius. These plots are also matching properly. This is the D infinity versus V, and it shows a sigmoid kind of curve and it increases with voltage. This is also matching properly. This is F infinity versus V. It decreases with increase in voltage, and it has also a sigmoid kind of behavior. This is FCA infinity. It also decreases, but it does not goes to zero. It goes to a constant value at higher voltage. This is the tau d values, and this solid line is the the model prediction, and which is also clearly matching here. This is tau vef. This is R infinity, S infinity. Here you can see it is for epicardial and endocardial, and these are also matching. So for different epicardial, endocardial, and myocardial tissues, the properties are different. So all these parameter can be different there. This is tau bar. It is also matching. Tau s for epicardial, tau s for endocardial, this is the current versus time plot, and it is showing. So whenever it is a voltage clamp experiment, so when a voltage is changed, since we saw this kind of peak behavior, and it is also matching clearly. This is another current plot. So hereafter it is a current voltage relationship for epicardial and endocardial. So we can see the slopes are different. So the net conductivity is different in both epicardial and endocardial at higher voltages. This is the XS infinity square graph. It also matches properly. This is tau XSS. This is IPF versus V. This is IV relationship for IKS plot, IKS current. These are also few parameters. These also match properly. Um, Hereafter, this in the seventh, eighth, and hereafter, we need an intercellular sodium and calcium current values. So these ionic concentration values for sodium and calcium inside the intercellular region are not provided. So that's why we can see so we assumed a different value here. So that's why the time scales at which this voltage drops is different here. So hereafter in few plots we can see some variations, but uh, the graph shows quality graphs are qualitatively similar, but the values are just different. Here also we can see some decreasing trend, but the values are different. That's because of some parameter values are not given in the paper properly. So all other plots match quite well here. Yeah, this is the APD versus DI curve, and here the values do not match properly, but we can see some trend like it increases sharply and then it gets flat. So that is captured here while reproducing the figures. This is the calcium stepping, but we were not able to do this properly here. This sodium stepping, the values are quite different here. Maybe the concentration values which were given in the paper are the unit is different, so that's why it is different. But the variation is very less in the reproduced plot compared to the actual plot. So it changes at third place, but here it changes at the second decimal place. Um, these are the voltage versus time plots for epicardial cell. So it shows action potential here, and this is for endocardial cell, and this is for myocardial cell. And this is the action potential APD versus APD distribution curve. So it is different in the paper for all the three cell types, but in the reproduced plot it is same. That because there is some error in reproducing the plot. So discussion. So what makes this different from the earlier model is that model successfully reproduce human AP morphologies, and the experimentally observed APD restitution behavior is reproduced here quite properly, and the conduction velocities were able to capture. So the main highlight this paper has compared to other animal based uh, heart models is that it considers slower sodium ion recovery compared to animal tissues. That's where the paper was able to capture this human heart ion behavior properly. So, coming to the reproducibility, the intercellular calcium and sodium ion concentration was not provided in the paper, so we assumed some value, and that's where the difference arises from. So, in the figure 7 to 10, where these values are important, a significant variation is there in the reproduced figure. Conclusion: figure 1 to 10 of this paper are successfully reproduced. The model proposed shows significant agreement with the experimental data, and due to some missing parameters, some deviations are observed in the figures published in the paper and the figures reproduced. Thanking you. So here is my Python code for this project. So first of all, I imported this all libraries. Then I defined all the parameter values as given in the table one. It follows around 90 equations in the paper. I defined each and every equation as different function, and these equations are first defined, and thereafter all these plots are generated. So to generate a plot, this kind of plot, we change the voltage values and calculate the corresponding change in parameter and plot the plots. So these are in the most of the plots. So and generate the to solve the ODs, as given in the paper, we use the Euler's forward method. So for some of the equations, there is the analytical solution is available. So we can see here it is some constant times change into exponential times t. So this analytical solutions of the equation is used to get the current versus recovery time plots here in one plot, and these are again some parameter variations with uh, voltage. It takes a lot of time, around 15-20 minutes to run, so I can't show every step as a live demonstration, but these are the results. Uh, yeah, so here we are giving a different pulse in EK, and seeing how the current changes. So it is a voltage clamp experiment here, and these are the plots for voltage clamp experiment, and these are the steady state current versus voltage showing the IV relationship for epicardial and endocardial cells. These are again some parameter variations with voltage. Um, this is current versus voltage for IKR. This is to simulate the action potential, we solved it using IVP. So there is no analytical solution available for this large system of equation. So we defined the ODE function, and we solved this using solve IVP method as shown in here. So So multiple peaks are given, and then the last one is shown here. And this is another one showing two different peaks. And here we use the the S1 S2 restitution protocol. So here we can see if it is uh, a, 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 a multiple of 1000, so then we give this S2 pulse, otherwise we give the normal pulse. So we were able to simulate up to 10th one, but after that the plots are for 2D spatial temporal variation, so that plots we were not able to solve. Yeah, thank you.